Lluis.
Thank you very much, Lucy. And also thank you so much for inviting me to give this talk. Obviously, I would have preferred to be in Liverpool, which I don't know, by the way, but at least this is a nice way to survive throughout this pandemic. So today, because I know that you are mostly interested in evolutionary anthropology, which also is my case, my background is population and evolutionary genetics, I'm not going to give you a talk of how and to understand the genetic basis of infectious disease susceptibility or things like that, but I'm going to give you a kind of population genetics and evolutionary vision of how we can understand indeed infectious diseases and immune responses between humans. Sorry. So what do we know over the last 20 years in human genomics to start with? So as you know, in 2001, the genome of the human genome was sequenced for the first time, and it cost like $3 billion. Today, sequencing a human genome 20 years after cost around 400, 500 euros. But most importantly, in the context of what interests us today, that is diversity, we know that there is no sense of talking about the human genome reference sequence. Because if you compare each of us, regardless of our ethnic or geographic backgrounds, to the human genome reference sequence, each of us will differ at 10,000 mutations that change the amino acid sequence. Each of us, we can have up to 300 genes that are disrupted, raising questions about the levels of redundancy. Also, each of us, we have in between 50 and 100 mutations in our genomes that have been already associated with an inherited disorder. The reason by which most of us were healthy is that these mutations are at the heterozygote state. And also, as you know, depending on the history and the demographic history of a population, there will be differences in the burden of deleterious mutations. In other words, like kind of classical populations, such as most African, most European, most Asians, that is, populations that are under demographic expansions, in average, will show lower levels of deleterious mutations than populations that have gone through strong bottlenecks or founder effects, such as Finnish population, Sardinians, Icelandic, French Canadians, etc. But a major question in human genetics more generally is what's the impact of these mutations on traits or diseases? So as I was telling you, there's kind of traditional approaches or more medically oriented approaches to understand the impact of genotypes on phenotypes, where you use patients basically. And either you apply clinical genetic approaches in which you have a family and you are going to follow which mutation or mutations segregate with the affected cases, or you perform genetic epidemiology studies like GWAS, genome-wide association studies, in which you have, let's imagine, 2,000 people with tuberculosis that you are going to compare with, well, 2,000 people with COVID-19, severe COVID-19, which you are going to compare with 2,000 people that have not developed a severe COVID-19. And best case scenario, which is the case, by the way, you are going to find 3-4 hits, like a region in chromosome 3, DABO blood groups, that seems to increase the risk of develop disease, in this case, COVID. But this is not at all what I'm going to talk about today, and this is not at all our expertise. Our expertise and interest is rather to work with healthy donors, meaning the general population, all of us. Because don't forget that we are the descendants of those people, individuals and populations that have survived past epidemics, the Black Death, the Spanish flu, tuberculosis. So somehow what we can do in population genetics is dissect today how this resistance is engraved in our genes because of the molecular signatures that are left by natural selection. So we know today that, as you know, that humans originated in Africa around 300,000 years ago, and that over the last 60, 80,000 years ago, they dispersed throughout the world. But during these dispersals, humans had to adapt to different climatic conditions, to different nutritional resources, but also to different pathogens. And this is the basis of host biological adaptation through natural selection. So you may wonder why we're interested in natural selection related to infectious diseases. Well, because who says natural selection says strong mortality. And as you can see in these survival curves here, infectious diseases have been imposing a strong burden of mortality basically until yesterday from an evolutionary point of view. As you can see here, the UK, well, Liverpool, by the way, it's not done on purpose, but it's a nice coincidence. Liverpool, 130 years ago, 40 years ago, sorry, just just 38% of the population is the yellow curved, reached the age of 40. Today in the UK, well, in the 2000, 98%, 99% of the population reached the age of 40. This is thanks to all what happened in the beginning of the 20th century, the development of antibiotics, vaccines, and the improvements in hygiene. So infectious diseases have been a major cause of mortality, evolutionary-wise, and as you can see, they keep on being a strong mortality factor, as you can see here in purple. Mozambique, the same year as the UK, just around 50% of the population reached the age of 40. So basically, we try to understand this. We try to understand how pathogens have exerted pressure on immune genes. But also, as you will see, we try to understand which are the factors that makes that all of us, apparently healthy people, were not equal in terms of our immune responses. Which are the factors that makes that some of us, we are here, that we have a very strong immune response, that may protect us against infection, but the price to pay for that is that also it can make us more susceptible to develop autoimmunity, allergies, inflammation, and why some of us, we are here. We have a weaker immune response. So in systems immunology also, or in population immunology, what we try to understand is which are the factors that drive immune response variation in humans. Genetics, age, nutrition, microbiome. You will see an example of that. So let me share today with you 3 examples. In the first one, I'm going to tell you about how we can use population genetic approaches to understand basically how pathogens have exerted pressure on immune genes. So for that, probably the most important slide is this one. Basically, in population genetics, we know, and probably you know it very well, that this is the past and this is the present. These are chromosomes and mutations. So this is the present, and in population genetics, we know how to distinguish genes that evolve under purifying selection or under other forms of adaptive evolution, such as positive selection or balancing selection, and distinguish them from genes that evolved under neutrality. I'm not going to spend a lot of time here because you are evolutionary biologists, you are not like MDs, but just a brief reminder, purifying selection is mutations here in blue are neutral mutations, meaning no impact on the phenotype. And purifying selection will basically remove these black mutations because they are deleterious for the individual. Whereas positive or balancing selection will increase in frequency the mutation of a beneficial variant. either following a classical sweep model, meaning this mutation here in red, let's say, protects against tuberculosis. So over time, this mutation will increase in frequency. Or one form of balancing selection is basically the advantage of being heterozygote. So how can we use this information to better understand immune genes that have been of major importance to fight pathogens? Basically, genes that evolve under a strong purifying selection are genes that cannot tolerate functional variation, are genes that when you dissect them molecularly wise, you see that there is a default of functional variation. That means that they are very strongly constrained. That means that when they do accumulate functional variation, this must be deleterious. That's why you don't see it. So genes evolving under purifying selection are genes whose functions are essential, and are genes that if they appear mutations in them, there might be good candidates to be involved in severe disorders. Whereas genes evolving under positive selection or balancing selection, in contrast with genes evolving under purifying selection, are genes where novelty or diversity is favored and whose functions and mechanisms have participated in human adaptation. So we have been applying this over the last years to innate immunity receptors. What are innate immunity receptors? Where you are infected by a virus like flu, SARS-CoV-2, or a bacteria or a fungi. You have cells that come with receptors, and these receptors are going to recognize that something strange to your body has infected you, basically. So here in color code, you have the intensity of selection acting on different groups of major innate immunity receptors. And these studies have allowed us, for example, to distinguish genes that are essential and non-redundant, such as endosomal TLR genes that are involved here in red, that are involved in the sensing of particularly nucleic acids from virus, and distinguish them from other genes like present high levels of redundancy, such as this gene, TLR5. TLR5, there is a mutation that is in around 15% of Europeans, 30% of Middle Easterners, that is a stop mutation. And this mutation acts in a dominant negative manner. In other words, what this means is up to 15% of Europeans who are knocked out for TLR5. This doesn't mean that all, and we live very happily without. So that means that TLR5 has a redundant function. In this case, TLR5 recognized flagellated bacteria. That means that when you don't have TLR5, there's other functions that will take over. So we have been applying this at the genome-wide level, and we have been identifying the 1% of most strongly constrained genes, meaning the most essential, and proof of concept that this also helps us to identify genes, mutations in which might be involved in severe disorders, is that mutations in some of these genes have been associated with severe pediatric disorders, therefore strongly selected because it happened before the age of reproduction, such as herpes simplex encephalitis or Mendelian susceptibility to mycobacterial infections. But as I was telling you also, we can identify positive selection, meaning mutations that have been strongly beneficial for specific populations. This has been the case on some molecules like our called interferons. Here, you may have heard about interferons because they are also involved in resistance to COVID. And 10 years ago, actually, we identified these 8 mutations in type 3 interferons that we predicted to be very advantageous for Europeans and particularly for Asians. And the proof of concept that we're identifying the right mutations is that at the same time, other groups showed that some of these mutations are involved in a spontaneous clearance of hepatitis C virus upon infection. So basically, over the last years, we have been identifying mutations in specific genes that have conferred specific selective advantages to African, to Europeans, and to Asians. Our approach not only tells us, well, these genes have been beneficial in Europeans in this case, but we can identify also the most likely mutations. So a question here that we address, well, since in population genetics, we know how to date the time at which positive selection started, let's take all these genes and all the mutations that we predict that are under strong positive selection in each population, and let's date them. This is what we did, and actually we found that in most cases, selection started in between 3,000 and 13,000 years ago. That, as you know very well, is the time at which populations in different parts of the world started to shift from being nomadic hunter-gatherers to being sedentary farmers. As you know, this major transition dramatically changed population sizes, expose humans to new pathogens through the cohabitation with domesticated animals, and all that probably modified the pathogenic cocktail to which humans had to adapt. But also, we have been quite interested in the admixture of humans with Neanderthals. And why do we care about Neanderthals in the context of immunity? Because around 10 years ago, there was a paper from Stanford, from the team of Peter Parham, that showed that some HLA haplotypes, meaning these are genes, for those among you that are not very acquainted with immunology, that participate also in immunity, basically. And they showed that some HLA haplotypes present in present-day Europeans comes directly from Neanderthal. We know today that around 2–3% of the genomes of Europeans and Asians comes from Neanderthal, reflecting an ancient admixture with this hominin. So here, the question we wanted to address was, if we take all innate immunity genes, that are around 1,500 genes that participate in our first line of host defense against pathogens, and if we determine if they are enriched in Neanderthal ancestry with respect to the remainder of the genome, this would support the hypothesis that when early Europeans came from Africa, around 50,000 years ago, early Europeans were not adapted to the pathogens of Europe, but they encounter a hominin, Neanderthals in this case, that they were living in Europe for at least 300,000 years. So therefore, it's highly likely that they were adapted. So the idea is that through admixture with Neanderthals, modern humans may have acquired adaptive mutations that allow them to better survive the pathogens they encountered. So we did that, and actually the answer was yes. Both in Europeans and in Asians, innate immunity genes are enriched in Neanderthal ancestry with respect to the remainder of the genome. Here you have an example of 3 genes that play a major role in recognizing particularly bacteria on the cell surface. Here there must be around, each line is one individual. Here are around 100 Europeans. In red shadows, you have the genomic position of the genes. And when you have a kind of dashed line, white, means that it's a modern human haplotype. But when there is a blue line, means that there is a Neanderthal haplotype. So you can say that up to 30% of Europeans, we have the whole genomic region covered by a Neanderthal haplotype. Now, at the functional level, we are trying to understand why incorporating in our genomes 50,000 years ago, these Neanderthal haplotypes was beneficial for our immune responses. And also, we have identified other genes participating in major antibacterial or antiviral responses that present among the highest levels of Neanderthal ancestry in modern humans. But we don't need to go so back in time in terms of admixture to ask about how adaptive has been admixture for humans in general. Because all of us, to some extent, we are admixed in a way or another. Just Europeans, we are the result of at least 3 different main populations: original European hunter-gatherers, Middle Eastern farmers, and then Eastern Europeans, herders associated with the Yamnaya culture. Not to mention other populations like the African Americans that are right the admixture between Europeans and Africans. So to evaluate how beneficial might have been in some cases admixture for better adaptation, we focus on Africa. And why in Africa? Because in Africa, as you know, around 3,000 to 5,000 years ago, occurred one of the major migrations, or at least cultural revolutions in human history. That is the expansion of Bantu languages from the area that is today in between Cameroon and Nigeria, associated with agriculture and iron usage. So during these migrations, Bantu languages were basically dispersed all over sub-Saharan Africa. But today, as you can see in the right, there's a few surviving pockets of hunter-gatherers, the rainforest hunter-gatherers of Central Africa, that are also known as pygmies, East African pastoralists, and Khoisan populations from the south. So the question we wanted to address here was how, during these migrations of Bantu speakers, first, to which extent they admixed with the local populations of hunter-gatherers they encountered. And for that, we analyzed around genome-wide variation of around 2,000 individuals from 57 populations of Africa. And I don't know if you are familiar with this kind of plots, admixture plots, but just in a second. Here there's 2,000 individuals. Each line here is one individual. And basically in different colors, you have different genetic components. As you can see, here are Western Bantus, Eastern Bantus, and Southern Bantus. So most Bantu populations, like 70, 80% of their gene pool is of red cluster, okay? But as you can see, Western Bantus, they have this green component. Eastern Bantus, this pink component, and Southern Bantus, the yellow component. And basically, from which populations they come from, easy. The green comes from pygmies, Western rainforest hunter-gatherers. Eastern Bantus admixed with East African pastoralists, and Southern Bantus admixed with Khoisan population. Khoisan populations that you can see here. But most importantly, what we showed is that this admixture has been sometimes beneficial. Western Bantus acquire, thanks to admixture with the pygmies, beneficial alleles located in the HLA region, so involved in immunity, and also in a gene called CD36 that is involved in resistance to malaria. And Eastern Bantus, through admixture with East African pastoralists, they acquire the allele in the lactase gene that allow adults to digest milk. So highlighting the beneficial nature of admixture for better adaptation. So now let's see, the second example I want to share with you today is to try to understand more generally the factors that drive immune response variation in humans. Try to quantify, to pinpoint the evolutionary factors, the genetic factors, and the environmental factors. So for that, how do we work? Basically, we work at the population level. We recruit individuals, either from the same population or from different populations, from whom we obtain their blood, and their blood is infected with different pathogens. And then we assess the gene expression before and after infection to evaluate how strong is the transcriptional response in this case to infection. And then we combine this expression data with genetic data from the same individuals to map the genetic basis of differential responses to infection. So to do that, we have been focusing in the last years into cohorts. Let's start with EvoImmunopop. What's EvoImmunopop? EvoImmunopop is around 200 individuals that we collected in Belgium, half of African ancestry and half of European ancestry. Why collected in Belgium? Because it was a way to homogenize as much as we could environmental exposure. Let's say to avoid Africans from Africa and Euros from Europe. In that case, from their blood, we purify monocytes. It's a major innate immunity cell type. And we infect them either with influenza virus or with different ligands that we called that mimic bacterial or viral infections. And then we map the genetic basis of variation in immune responses through expression quantitative trait loci. What are EQTLs? I guess you know it, but just for those that are not familiar. An EQTL is a mutation that is basically associated with changes in gene expression. This mutation is not associated with any change in gene expression, as you can see. So this is not an EQTL. Sorry for that. Oh, God. I'm sorry, I have a problem with my mouse. In the middle, this is an EQTL, because depending on the genotype, you are going to have more expression of the gene. And we are particularly interested in the last case, what we call response EQTLs, meaning the mutation has no effect on gene expression at the steady state. But the same mutation have an impact on gene expression when you expose cells to a stimulus, in this case, infection, highlighting gene-environment interactions. So basically, this is what we did. And here you have a principal component analysis where based not on genetics, on gene expression upon infection. And as you can see, most of the variance is just explained by the condition of a stimulation. Here you have the non-stimulated individuals. Here you have the individuals exposed to influenza virus. And here you have different viral and bacterial ligands. But if you zoom in one of the conditions, for example, let's take the example of TLR1 and 2, this mimics responses basically to bacteria, you can start to see difference also between populations. And basically what we found, to cut a long story short, is that obviously Africans and Europeans don't respond in an on-off way, meaning very different, but we found marked differences in the intensity to which they respond to infection. And again, to cut a long story short, we found that in 80% of the cases, these differences in the intensity of immune responses can be attributed to EQTLs, meaning mutations that regulate gene expression that are simply differently distributed at different frequencies between Africans and Europeans. Sometimes, as an example, a single mutation can explain strong differences in responses to infection. For example, a mutation in this gene that is called TLR1. This mutation is present only in around 1% of African, but in around 70% of Europeans. And this mutation is associated with a reduction of the inflammatory response accounted for around 100 genes. In other words, the 70% of Europeans that we have these mutations, we have an attenuated immune response. And moreover, we have found that this mutation presents strong signatures of positive selection, suggesting and highlighting the compromise between, well, recognizing a pathogen, but not exaggerating the immune response, because certainly you are going to neutralize the pathogen, but also the exacerbated inflammatory response is going to be harmful for the host. This is the case, for example, in the case of severe COVID. What I'm sure you've heard about these cytokine storms is that the inflammatory response is so strong to get rid of the virus that basically yourself are provoking a severe manifestation of the disease. And also, to finish with this example, we have found that in general, EQTLs, again, mutations that regulate gene expression in monocytes in this case, are enriched in Neanderthal ancestry, and particularly mutations that are involved in antiviral responses, suggesting that through admixture with Neanderthals, early Europeans acquire mutations that allow them to better survive viral infections. This study has been confirmed by another study 2 years later by Stanford people that they showed that VIPs, viral interacting proteins in humans, are enriched in Neanderthal ancestry. And this is just to show you also that we have been analyzing other layers of gene regulation, like enhancers, promoters, microRNAs, and we have found basically that enhancers are enriched in Neanderthal ancestry, but particularly those that are active in fat tissue, which raises questions about adaptation to cold, and in T cells that are also a major cell type related to immunity. Let me share you also with you an example of another project that is called Milieu Intérieur, our internal milieu. So here we adopted basically the opposite strategy that EvoImmunopop. Here we wanted to reduce genetic diversity. We were not comparing African with Europeans or with Asians, and we just focused on Western European people. We wanted to reduce as much as we could genetic diversity. They were collected in Brittany, here in France. So, but we wanted to take into account many other parameters, lifestyle parameters, microbiome, etc. So for that, we recruited in that case 1,000 people in Rennes, Brittany, stratified by sex and age, from 20 to 70 years old. So in 5 groups from 20 to 30, 30, 40, and so on. Half men, half women. From each individual, we took blood, and their blood was stimulated with 30 different microbes or molecules that activate the immune response. And most importantly, from each individual, we have an extensive case report form where we have socio-demographic variables such as, well, income, education, etc., vaccination, medical history, sleep habits, nutrition, and physiological measures to have a much more comprehensive idea of the different factors that can drive immune response variation. So let me share with a few examples just. The first question here was, if we take the whole blood of all of us, there are differences between the immune cell types that there are in our whole blood. So among the 140 variables that may explain this variation, age, sex, genetics, nutrition, depression, sleeping, blah, 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 until 140, which are the factors that drive variation in immune cell types in our blood? So out of the 140, basically we found that just 5 factors have a major impact. The genetics of the host, chronic CMV seropositivity. CMV stands for cytomegalovirus. Many of us are infected by this chronic virus. We don't even know that. Age, smoking status, and sex. And basically, forget about this complicated plot, but basically what we show is that while genetics, here in blue, will mainly affect variation of innate cell types, non-genetic factors, are going to affect, like sex and age, variation in adaptive cell types, the ones that takes the memory, basically. We did exactly the same basically which are the factors that affect microbiome diversity in the 1,000 people. So for that, we did shotgun sequencing from their stools, and we associate all these different species of bacteria, to the different variables. And basically we show, that 15% of the species in our gut microbiome are dependent on sex, and 40% of the species are differentially abundant depending on age. And also, well, we found other variables that explain a bit less, but they also explain variation in our gut microbiome. And finally, the last example I want to share with you, all what I've been telling so far for this study of milieu intérieur was at the steady state, our microbiome, or cellular diversity in our blood. But what about when we infect blood? So this is what we did in the 1,000 individuals. We use different bacteria, viruses, or fungi, as you can see here. And again, here you have a principal component of the expression data: non-stimulated, influenza virus, and other bacteria and fungi. And the question was, which are the factors, again, that explain this diversity? And what we show is that 20% of the variants in transcriptional responses to infection are basically explained by our natural differences in different immune cell types in our blood, followed by genetics, here, acting in cis or in trans, followed by sex and age. Of course, this is an average across all immune genes. There are some genes whose response to infection is explained, 60% is explained by genetics, and other genes whose variation is mainly explained by sex or age. But still, a large fraction of the variance in immune response variation remains unexplained. And now we are following up this using single cell technologies. Basically, we are taking cells from people from different sex, different age, even different BMI, body mass index. And at the single cell level, we infect these cells with influenza virus, for example, but also with SARS-CoV-2, because as you know, sex and age are major risk factors in order to try to understand how these affect single cell responses to infection. And to finish the last 5 minutes, I want, because it's really quick, I want to share with you 2 examples of how, using population genetics, we can learn more about specific diseases in specific populations. The first is malaria in Africa. And why here? Because we wanted to revisit the most iconic case of balancing selection in humans. We know it from the 1950s, a British guy, by the way, called Anthony Allison, that he showed that if we are, as you know, heterozygote for the sickle cell allele, we are protected against, at least higher protected against falciparum malaria, despite that the homozygote state would develop sickle cell anemia. So our rationale here was, well, in the absence of malaria, meaning there is no selective advantage of being heterozygote, the sickle cell allele will be purged from the population, will be eliminated from the population in just 12 generations because of the deleterious nature of it. So the rationale here was, well, let's date the time at which selection started, because it's a way to date the time at which malaria started to be an important burden. So for that, we sequenced the hemoglobin gene in around 500 individuals of hunter-gatherers and farmers from Africa. And what we found basically was the sickle cell allele using ABC, approximate Bayesian computation. The HBS, the sickle cell allele, originated in the ancestors of present-day agriculturalists, the so-called Bantu speakers, because 5,000 years ago we're not farmers, we're hunter-gatherers. So pushing back of a few years, the putative origins of malaria in Africa, but most importantly, what we found is that hunter-gatherers acquired the sickle cell allele only in the last 6,000 years. This is what supports what's called the poison antidote hypothesis, meaning I'm a farmer, I start to migrate, I start to do deforestation, I bring therefore malaria to the hunter-gatherers, the pygmies in this case, but I not only bring in malaria, I'm also bringing the antidote, that is the mutation that allowed them to resist malaria, that is the sickle cell allele, highlighting again the adaptive nature of admixture. And the last example I want to share with you is a recent one unpublished so far that is about tuberculosis in Europe. As you know, tuberculosis is still one of the big three killers in the world, together with HIV and malaria. And despite in Europe, as you can see here, we are doing much better. Tuberculosis is supposed to have been the deadliest infectious disease throughout European history. And until a few years ago, actually, we knew very little of the genetic basis of susceptibility to tuberculosis in humans. And a team in Rockefeller University identified the first mutation that is common in the population, common meaning 2.5, watch out, this is not 25, this is 2.5 frequency. And this mutation is present in around from 2 to 4% of European people, or people that are known to be admixed with Europeans. So as you can see, from 2 to 4% of European ancestry people are millions of people that have this mutation, and this mutation increases the risk to develop tuberculosis. So what we wanted to do here was to understand the evolutionary history of this mutation. And what we did for that was to take advantage of a public database from Harvard of 1,000 ancient individuals that had been living over the last 10,000 years of European history. So here, you have the frequency of the mutation in Europe. You see that the mutation appeared for the first time during the Mesolithic, around 9,000 years ago, then increases steadily in frequency to almost 10% during the Bronze Age, to then decrease dramatically in frequency from the Iron Age on until the present day 2-4% frequency. So in population genetics, we can say, well, this decrease in frequency, is this due to random processes, drift, or is this due to negative selection? Because if this is due to negative selection, this means that the mutation has started to be under strong negative selection in the last 3,000 years, which will tell us that probably tuberculosis was important. So what we found is that this mutation actually originated in West Eurasians, Europeans basically, 30,000 years ago. But most importantly, what we found is that the mutation started to evolve under strong negative selection only in the last 2,000 years ago, with a selective coefficient among the strongest in the human genome, in that case, negative. In other words, these studies suggest that tuberculosis have imposed a strong burden on European health in the last 2 millennia. And with that, I would like to thank the people of my lab, our collaborators, funding agencies, and you for your attention. Thank you very much. I'm almost in time.
Thank you, Lluis. That was very, very interesting. If you'd like to just stop sharing your screen, we'll go into the answers session. Hopefully my connection is okay, but if not, Karis will take over. Okay, so we have a few questions already. What do you think about potential recent relaxation of natural selection, and in what way can this question be addressed?
Certainly in some parts of the world, for some specific functions, there must have been some relaxation, meaning when you start to use, well, you saw the data of Liverpool in the end of the 19th century, just 38% of the population reached the age of 40. Today, 98, 99 of British people reach the age of 40. So certainly vaccines, antibiotics, and hygiene in the context of infection have certainly relaxated the selective constraint. Well, this doesn't mean that there's no other problems like antibiotic resistance, for example, that appear. However, to address this, I always like to say, well, set up a population genetics group in 4,000 years, and you will see that something major probably happened in the 20th century that modified the course of natural selection in some parts of the world. Don't forget that in many parts of Africa, many parts of Southeast Asia, natural selection is really still brutal, right?
Okay, so next question from Nicole. My question is a must. What is your opinion about the apparent relationship between Neanderthal ancestry and COVID-19 risk?
Okay, that's a very interesting and timely question. There are several examples. Well, not only is the question is interesting, but also allows me to say something that I didn't say in my talk. Don't forget that things that have been beneficial in the past are not necessarily beneficial today, because natural selection doesn't know that the environment can change. And there is, now I will talk about Neanderthal just a second, but we have increasing evidence of mutations that are under strong positive selection, and the same mutations are associated with increased risk to develop autoimmunity and inflammation, which sounds super counterintuitive, right? How is it possible that a mutation is under strong positive selection if it increases the risk of a disease? The answer is just look at what diseases. So there are examples, like in celiac disease, for example, that the same mutations that protects against infection are associated with increased risk of autoimmunity. And this is this kind of trade-offs. It's much less robustly supported, but in the context of metabolism in the Pacific, there's also cases in which apparently some mutations that are under strong positive selection are associated today with metabolic diseases such as diabetes or obesity. And is all this theory of the thrifty genotype hypothesis, meaning mutations that all over evolution has been positively selected because we have been living in famine for all of our history, basically. And with the minimum amount of food, you maximize it, the energy intake today, with the availability of food, they are the cause of obesity. It's a bit less supported. To come to Neanderthal, yes, there's a region in chromosome 3 that increases our risk to develop severe COVID and has been inherited with Neanderthal. My hypothesis about that, because there's no functional data for the moment, is that this region is among the many other regions that increase the inflammatory response. Because evolutionary speaking, having a very strong immune response has been something good. But today, actually, this mutation is associated not to increase risk of being infected. This mutation is associated with increased severity of being hospitalized, meaning that you have a strong inflammatory response. So that's my opinion. And by the way, there's another paper that is not published yet, but is in bioRxiv of the same group that have found another gene that is inherited from Neanderthal, that in that case protects against COVID. So it's not black and white, all this, yeah.
Interesting. Okay, next question from Tim. Can you reconstruct the rate of mutations we see over all of time? Does this identify time periods where there are higher or lower rates of mutation, and how do these relate to known past events or allow you to infer the frequency of, e.g., past pandemics?
Okay. I think, just to be sure that I understand the question, because there's two parts in that question. There is there particular periods where there is just more mutations? This is why I understood. Well, mutation rate, to my extent, to my knowledge, doesn't change so much over time. However, there can be more mutations because of demographic events, such as, for example, we do see that in the end of the Bronze Age, in general, there is an increase in diversity in the European population. And this is because we know that right at that moment, people from Eastern Europe, from the steppes, came through the Yamnaya culture. In that case, we can pick up a demographic event. The second question, which is very interesting because it's actually what we're doing now at the genome-wide level, is there particular periods over the last 10,000 years of European history in which some mutations in immune genes have massively increased or decreased in frequency, which could give us some hints of particular moments of adaptation to infection, either adapting favorably or removing mutations. We don't know it yet. This is what we are doing in the moment, trying to understand this at the genome-wide level. Yeah.
Thank you for that question. So we do have time for some more questions if anyone has any more.
Yep.
Thank you.
Well, if somebody's curious and shy, they can send me an email and I will answer.
Okay, well, I guess if there are no more questions, then we can finish up early. So thank you very much, Lluis, for that great talk. And thank you, everybody, for coming. And we'll see everybody next week.
And thank you for the invitation and for everything. Ciao.
Bye.
Bye-bye.